QIAGEN.
Sample to insight.
As already introduced, I'm working in the clinic and I'm a biologist, and so I'm closely working together with the medical doctors who introduce the different types of gynecological cancers to us and talk about the main problems in these malignancies. So we do a lot of research in breast cancer and in ovarian cancer, and today's topic is ovarian cancer. And ovarian cancer is the fifth leading cause of death in women who have cancers. And the problem in ovarian cancer is that it is diagnosed in very late stages. So all the women go to the gynecologist quite regularly. These tumors are detected in more or less stage 3, and this is an advanced stage. So within the next 2 slides, I will show you some images taken during surgery. So for those of you who cannot stand these kind of images, please close your eyes for some seconds, and then I will move on with the research data. So this is the initial situation in ovarian cancer, and you see that there's a really large distribution of tumor material. It's a very long surgery, taking in Germany about 8 hours. So the classical prognostic factors are tumor size, the age of the patients, grading, the histological subtype, and the most important factor is the tumor rest after surgery. And so this is the image after surgery. This is a complete tumor resection, and this is the main goal in ovarian cancer. And this residual tumor rest after surgery is the only influenceable prognostic factor in ovarian cancer. And so what is the strategy? So you see here how important this residual tumor rest after surgery is with regard to progression-free survival and overall survival. So you see that those patients who do not have any tumor rest after surgery have the best prognosis with regard to progression-free survival as well as overall survival. So this is the first step in ovarian cancer, that you really try to remove all the tumor rest and then start with the therapy. So what kind of therapy can we offer? So after surgery, all the patients receive platinum-based chemotherapy. It's in contrast to breast cancer, where you have a lot of therapeutic options. In ovarian cancer, platinum-based chemotherapy is the only therapy which has been working for decades now, and there's no other chemotherapy which is better than platinum-based chemotherapy. Of course, there are a lot of ongoing studies. At the moment, patients are treated additionally or in a combined treatment setting with Avastin, so anti-angiogenesis, or with olaparib. This is a PARP inhibitor, but only patients with BRCA1 and 2 mutations benefit from this kind of therapy. Now, the biggest problem in ovarian cancer is platinum resistance, and about 15 to 20% of the patients do not respond to that chemotherapy, but this can only be assessed in the follow-up of the disease. So we have no suitable marker at the moment to predict prognosis and platinum resistance. So the first question is translational research able to find predictive markers, on the one hand for progression-free survival, PFS, overall survival, OS, or platinum resistance, PR? And the second important question is, what kind of biomaterial is best to look for predictive markers? Is it the primary tumor? Is it blood? Is it bone marrow? And the next question is, what are the right markers? So what about the material? So the biomaterial, you can, of course, analyze a primary tumor. This has been done for LOH analysis, for next generation sequencing, but the problem is you have the primary tumor at the beginning of the disease, so at primary diagnosis, and you cannot use the tumor for follow-up of the patients. So on the other hand, it is well known that there's hematogenous tumor cell spread also in ovarian cancer, like in breast cancer. And we have different circulating tumor cells, which leave the tumor, move through the blood vessels, and some of these cells are able to move into the bone marrow as a secondary organ. And these cells are called disseminated tumor cells. And on the other hand, of course, we have circulating biomarkers in blood, mainly from these circulating cells like tumor DNA, microRNAs, and other biomarkers. But today's focus should be on CTCs and DTCs. So the questions that have to be answered: do we have reliable methods for selection and detection of DTCs and CTCs? And this is also what the medical doctors ask, because if you take patient material, you have to inform the patient. You need a votum of the ethic committee. The patients have to give their informed written consents. And of course, the patients want to have the results. And the next question is, are these cells of prognostic significance? And the third question, what are the characteristics of these cells? Because in oncology, you want to move into targeted therapy, and then you have to know the target you are targeting. So what about the methods? In bone marrow, we are quite lucky that we have something like a so-called standard method. So during surgery of the primary tumor, we make a bone marrow aspiration. We take bone marrow from 2 different sites. And this bone marrow is picked up by the laboratory, and we perform a density gradient centrifugation. And we isolate the mononuclear cells. You see this white layer. And within this white layer, we find the tumor cells. They have the same density. We isolate these tumor cells. And what we do is we do immunocytochemistry for the detection of these cytokeratin positive cells. And as you can see, after neofuxin staining, you can see red cells. Sometimes you can see clusters. And we have an automated system, the aerial, for the evaluation of DTCs, so that you can analyze 50 slides overnight. And afterwards, you see the little images and can verify whether you have a tumor cell or not. Now, if you look at the literature and look for publications dealing with DTCs at primary diagnosis, there are not so many papers published, so only a few of them. You can see the number of patients analyzed, you can see the positivity rate, and in most cases you see the same method has been used. And so the prognostic significance of these cells has been shown. And this is work from our group. This is our first publication on DTCs in ovarian cancer. And even analyzing only, let me say, 62 patients before and after therapy, we could show that those patients with an enhancement of DTCs after therapy, that they had a worse outcome. Of course, 62 patients is a small number, but with regard to ovarian cancer, you do not have so many patients a year like in breast cancer, but we thought we have to verify that in a higher patient number or in a bigger patient cohort. So the next thing we did, we asked other groups who did the same method and had the same patients. So we put all our data together and analyzed the prognostic significance of these cells. And this is a study which has already been published in 2013. This is the analysis of 495 primary ovarian cancer patients. And analyzing this cohort, we could show that DTCs present at primary diagnosis significantly correlate with a worse outcome. And in a multivariate analysis, DTCs were an independent prognostic factor for progression-free survival and overall survival, but not for platinum resistance. So the next question was, is the negative prognostic impact of DTCs related to a DTC persistence after platinum-based chemotherapy? and or to a cellular phenotype being associated with stem cell character, because there were some publications that stem cell factors like Lin28 or SOX2 are associated with ovarian cancer. And this is a study which has been published very, very recently, so only some weeks ago. And this is a study where we analyzed the bone marrow before and after chemotherapy. You might say, okay, but I think to the best of my knowledge, we are the only group who has bone marrow aspiration after chemotherapy. You have to consider that, for instance, for breast cancer, in case our patients are DTC positive, we offer a bisphosphonate therapy, which is quite successful in eliminating these cells. And this has also recently been published by our group in scientific reports. But for ovarian cancer, in case of DTC positivity, no clinical or therapeutic consequences are drawn. So the patients who give their bone marrow simply give it for research purposes, and to give bone marrow after chemotherapy, so not during surgery where they do not feel any pain because they are more or less sleeping. This is quite a rare event that you can have bone marrow after chemotherapy, so thanks a lot to the patients. And in total we have 79 patients at primary diagnosis where we could analyze the bone marrow before and after chemotherapy. So the study has already been published, as I told you, and this is an open access journal for those of you who want to go into detail, look at method establishment, all the details. Please feel free to pick a copy. So what did we do? So we had 79 patients, and of course we analyzed the prognostic significance of these cells with regard to progression-free survival and overall survival. And although this was a smaller cohort, we could show that the DTCs before therapy were of prognostic significance with regard to overall survival. If you look at the pairs, so at those patients who had a persistence of DTCs, the prognostic significance so there was borderline significance. And what was the most interesting result, I think, is this result, that we had patients where we did not find any cytokeratin positive cells, so we simply look for epithelial markers in the bone marrow, but we had a lot of positive cells after therapy. So these are the Kaplan-Meier curves showing you these numbers in figures. And the next step was that we said, okay, we look at the patients who had bone marrow positive cells after therapy. And of course, we had to do this analysis on additional slides which were left in the freezer. And we said, to see an effect or to have an image of the number of positive cells, we only analyzed patients where we found more than 5 cytokeratin positive cells to be sure that we will also find cells on the residual slides. And as a stem cell marker, we chose Lin28 and SOX2 because they were discussed to be relevant in ovarian cancer. There's also OCT4, which is important in ovarian cancer, but due to the number of slides available, we decided to analyze these 2 markers. So I will show you the results. So this is 4-color staining. On the one hand, under A, we have the DAPI staining. Then under B, we have a staining for cytokeratin. And under C, you see the red staining for Lin28. And of course, under D, you see that we also established a staining for CD34 because in the bone marrow, you find about 1.5% normal hematopoietic stem cells, and of course we had to exclude false positive results. And we also established a staining for CD45, also to be sure not to detect lymphocytes. And in the image E, you see then a Lin28 cytokeratin positive cell in the bone marrow of a patient after therapy. Quite interestingly, we also found cells that were Lin28 positive, CD34, CD45 negative, and also cytokeratin negative. And comparable results were also obtained for the stem cell marker SOX2. So to summarize that, the key message in this paper is that DTCs present after therapy express stem cell associated proteins. And DTCs with stem cell character were also detected in some patients where we found no DTCs before therapy, you know. So the first step was only to look for cytokeratin positive cells, and there were patients who were negative before and positive after therapy. But taking also analyzing these slides, so these patients we analyzed after therapy, we also took the slides before therapy and looked for stem cell-like markers. And in those patients where we did not find any EpCAM or cytokeratin positive cell, there were very, very few cells that were stem cell-like. And we believe that these cells progressed under therapy because these cells cannot be eliminated by chemotherapy. And this might also be the reason why the outcome of patients who changed from DTC negative to DTC positive was worse. And the fact that we also detected Lin28 or SOX2 positive cells that were cytokeratin negative, so we concluded that perhaps we might have two different cell populations. On the one hand, some kind mixture of epithelial cells and cells that had undergone epithelial mesenchymal transition. So what we can conclude that there are additional therapeutic regimens that are necessary to eliminate these cells. So of course, if you want to do more or know more about the cells, of course it's quite difficult to take bone marrow as biomaterial because it's very difficult, of course, to convince the patients to give the bone marrow after therapy or even longer at different time points. So we thought that a blood-based biomarker would be of more benefit. And this is why we started with the circulating tumor cells. We had quite good experience in the breast cancer setting. And so the main question is, what is the right method? So for the bone marrow, we were lucky to have a standard procedure. This method was already established in the 90s and is still the method of choice. But for the circulating tumor cells, I think there are more than 40 different methods for CTC selection available now on the market. And of course, everybody has a different goal for CTC selection and detection, and we working in the clinic with a high number of samples coming in every day, of course we need a high throughput method. And besides the prognostic relevance of CTC counts that has been demonstrated, I think predictive markers are needed. And in general, I think the community is moving from cell counting to the characterization of CTCs or a combination of both methods. And if you want to use CTCs as a liquid biopsy, of course, you have to know the target. And I think for this purpose, a characterization of CTCs is necessary. Now, when we started, it was just by chance that I started with a molecular method because in 2004, I tried to analyze the CTCs as I did for the bone marrow, and I took another enrichment step. And for breast cancer, we had about 4% positivity rate, and this was very low, and this was very expensive. And I thought, well, does it make sense to look for circulating tumor cells? And it was by chance that this ADNA test was introduced to our group. So we started in 2006, and over the years together with the researchers from that company, I think we developed quite good strategies to select and detect circulating tumor cells. So let me shortly introduce the test procedure. You do an immunomagnetic cell enrichment, and what is quite important that you use more or less a cocktail of different antibodies. So you know that some systems only use EpCAM as a marker, and And although EpCAM is expressed on the primary tumor in ovarian cancer, it's quite astonishing that you rarely find EpCAM on the circulating cells. I know an Austrian group who started with a cell search and tried to select the CTCs for ovarian cancer only with EpCAM, and they were not quite so successful. They had 4% positivity rate. So whatever you're doing using antibodies for selection, I think it's necessary to use a cocktail of different antibodies in order not to lose too many cells. And is that you do mRNA isolation and reverse transcription, and this is followed by a multiplex PCR. Well, this is the whole procedure, how it is working. So you take the blood, incubate the blood with the magnetic beads. This is so for about half an hour, and then you put them at a magnet, select the positive bound cells. You lyse the cells, you do RT-PCR, the multiplex PCR, and from the start, from sampling to detection, it's about 5 hours. Of course, you can interrupt after lysis of the cells. You can put them into a freezer. Within the next 2 weeks, you can do cDNA preparation and store the cDNA and prepare the PCR whenever you want to. So what is quite important, there are now different tests on the market. And every test uses different antibody mixtures, and this is optimized for the different tumors. So this shows you the different tests that are on the market. We have a lot of experience with the breast cancer test and with the ovarian cancer test and with the EMT and stem cell tests. And you see for the ovarian cancer what markers are detected. This is EpCAM, MAC1, and the antigen 12.5, and this is determined in a multiplex PCR and in a separate PCR when you can take the cDNA and look for ERCC1. This is a resistance marker. So what about the prognostic significance of CTCs in ovarian cancer? If you look at the literature, there are also not so many papers available. And so it started in 2002, and you see the number of patients, and you see the different methods that were used, and you also see the prognostic significance of these cells. And these are the papers using multi-marker gene panels, and you see using this method, you see a prognostic significance when you analyze circulating tumor cells. And this is a paper from our group published in 2011. And quite interesting, we were using the breast cancer setting for ovarian cancer patients because this is also an epithelial tumor. And we said, well, why not try and use this test also for ovarian cancer? These were our first steps. And as you can see, using this test, we saw that CTC is present before surgery and also after surgery, significantly correlated with a shorter overall survival. Of course, we said, well, we have to adapt that to ovarian cancer, and then, the test, which had been then constructed afterwards, included the ovarian cancer antigen 12.5 and GA7332. This is synonym for EpCAM and MAC1. And so we made the next step and analyzed patients using this test. And this paper has been published in 2014 by one of my former PhD students. And you see the number of patients, 143 patients at primary diagnosis of ovarian cancer. And those patients with circulating tumor cells at primary diagnosis had a worse outcome with regard to overall survival. And then we said, well, we also have patients where we have the CTCs after therapy, and analyzing patients and saying whether, well, positive before or after surgery, see there is a big difference in patients who are CTC positive and CTC negative, and those who are CTC positive, they had the worst outcome. Now, the hypothesis was, or the question, is the negative prognostic impact of CTCs perhaps arose it from a cellular phenotype being associated with platinum resistance? We already knew from our bone marrow results that stem cell-like cells play a role, but our main goal was to look for platinum resistance. And so what we did, we took the test ovarian cancer, and in a singleplex PCR, we analyzed ERCC1, which is one marker for resistance, often analyzed in a primary tumor tissue. So we evaluated the prognostic significance of ERCC1 positive CTCs. And that means the patient had to be positive for the ADNA test itself. So one of the markers, EPCAM, MAC1, and CA12-5 had to be positive. And additionally, the patient had to be positive for ERCC1. And doing this analysis, you can clearly see that patients with CTCs that express ERCC1 have a shorter progression-free survival as well as a shorter overall survival. And we were also able to do multivariate analysis, and this was really a fantastic result showing that ERCC1 positive CTCs were an independent prognostic factor for progression-free survival, and most importantly, it was an independent factor for platinum resistance, so for clinical platinum resistance. So to summarize that, the presence of ERCC1 positive CTCs, potentially platinum-resistant CTC subgroup, is an independent predictive marker for primary platinum resistance and poor prognosis of ovarian cancer patients. So what are the next steps? Let me share some brand new data with you, which were presented during this year's ACR in New Orleans, and these data will be shortly submitted for publication. So ERCC1 expressing circulating tumor cells, can we use them as a potential diagnostic tool for monitoring response to platinum-based chemotherapy and for predicting post-therapeutic outcome of ovarian cancer patients? So first of all, the question was, is the additional analysis of ERCC1 able to influence the overall CTC detection rate? Because what we saw right from the start, that we had patients who were negative in the ADNA test, but positive for ERCC1. And so because to present quite clear data, we said, well, we only say positive if they are double positive. So positive in the ADNA test and in addition positive for ERCC1. But of course, you have different cell clones in the population. It might be the case that cells lose a character and become only resistant. So this was the first question. Can we perhaps enhance or influence the detection rate, including ERCC1, into the ADNA test? And so performing a paired analysis, so before and after the therapy, we inquired whether ERCC1 positive CTC dynamics mirror response to platinum-based chemotherapy. So we had 65 ovarian cancer patients where we were able to study blood before and after therapy. This time, again, we said CTC positive. At least one of the three markers of the ovarian cancer test has to be positive, and ERCC1 again was analyzed in a separate PCR, but we made different evaluations. So let me walk you through the data. If you look at the first two columns, this is, let me say, the old classical ADNA test ovarian cancer, where we detected CTCs in 28% of the patients before and in 21 patients after therapy. So in the next evaluation, we said, well, positive is a patient who is either positive in the ADNA test or ERCC1 positive. And taking or including ERCC1 in the analysis, we could enhance the detection rate to about 40% before and after therapy. And then we said, well, let us only look at ERCC1. And we could demonstrate that we have a group of patients who only express ERCC1 in the cDNA samples, 14% before and 19% after therapy. So there's even an enhancement of detection. And then we took the old classification from our first publication in 2014, saying, well, we analyzed patients who show a double positivity. So the ADNA test has to be positive and ERCC1 has to be positive. And you see comparable results that we had 18% positives before and 13% after therapy. So as already published in 2014. Now, what about this special group of patients? We have patients, though 75% about these patients who do not express any marker on the cells. But we have a group of patients who only express ERCC1. And these are 90% of the patients before and 24% of the patients after therapy. And if we look at the double positive patients, you see that 75% of the patients never show any markers on the cells. But there are 5% of patients with a persistence of cells and 14% of patients where cells are eliminated after therapy and 8% of patients who change from negative to positive. So what is the prognostic significance of CTCs and ERCC1 positive CTCs with regard to progression-free survival and overall survival? So, the first two figures show you ERCC1 positive CTCs, and you can clearly see, looking at the blue and the green line, that those patients with the persistence of cells, or those patients changing from negative to positive, have a shorter progression-free as well as overall survival. And this is highly significant. And only looking at ERCC1 positive patients, you see the same results. Also, again, those patients with the persistence of these cells and patients who change from negative to positive have a shorter progression-free as overall survival. And we also see a significance for platinum resistance. So, to summarize these brand new data, the additional assessment of ERCC1 transcript expands the spectrum of CTC detection and defines an initial highly overlapping fraction of ERCC1 expressing CTCs, which are potentially selected by platinum-based chemotherapy. And we also suggest that besides their capacity to predict platinum resistance at primary diagnosis, ERCC1 positive CTCs could additionally be useful as a surrogate marker for monitoring response to platinum-based chemotherapy and to assess post-therapeutic outcome of ovarian cancer. Now, what is the outlook? I think with the knowledge we have now, we have to establish a multi-marker gene panel to explore the heterogeneous CTC population in ovarian cancer, because looking at the brand new ASCO studies, they show that ovarian cancer is now also moving into targeted therapy. And to move into targeted therapy, we have to know the target. And our gene panel should include epithelial markers, stem cell markers, and markers that indicate an epithelial mesenchymal transition. And that this procedure is successful, this has recently been published in Oncotarget for our breast cancer patients. And I think this is a very good and interesting paper. The work has been performed by my PhD student Maren Bredemeyer, who analyzed metastatic breast cancer patients at the time of progression and in the follow-up of the disease, and used a multi-marker qPCR gene panel, so quantitative PCR, and this over therapy. And you can nicely see how cells can be eliminated by targeted therapy. So this is also open access. So if you are interested, please feel free to take a copy. Well, I would like to thank you very much for attending today's webinar. So if you have questions, please don't hesitate to contact us. Thank you very much. Bye-bye.
Thank you very much, Professor Sabina Kazimierz-Bauer, and for very nice presentation, and also thank you very much for sharing your research data with the audience today. And to all the audience, if you have any questions, please feel free to type in the questions box, and we'll answer your question, okay? So if you have to go, also you can write down the email address. We are showing here, QIAwebinars@QIAGEN.com. So you can contact us at any time, and we'll answer your questions, okay? And I have one question about antenna test. So what have you ever, there are a couple of circulating tumor detection products in the market. Have you ever tried the other case? And how do you feel this antenna test in your research?
So I tested density gradient centrifugation, so using the OncoQuick system, and this has not been successful. We had a positivity rate about 4%, and I know that another group has tested the parasitics with some ovarian cancer cell line, and the recovery is also not convincing.
Thank you very much. So that I also have another question regarding the sampling. So when you receive the surgery sample, what do you think the time? And some people think like different time you get the tissue sample or blood sample. Does this have any impact?
We have the advantage that our lab is in the clinic, so whenever blood samples are taken, we immediately pick them up, and so we process them immediately or within the next 4 hours. Of course, after an 8-hour surgery, we have the opportunity to use these special preservation tubes supplied by the company, but blood in our setting is always taken 1 day before surgery or closely before surgery, that this does not interfere with anesthesia or so. And I think freshly prepared samples give the best results, although you can analyze samples 1 day later, but then you have to use the preservation tubes.
Thank you very much. And there is another question, very specific, ask the EMT. So when do you think the EMT will happen? Is during the dissemination from primary tumor into blood or during the circulation process?
I think this is immediately after leaving the primary tumor, because we have results for breast cancer, so very early breast cancer patients, and when we analyze these CTCs, we have a positivity rate about 15 to 20%, and a small amount of these CTCs shows these EMT characteristics. So when the cells leave the tumor, I think they even perform EMT right from the start. There might be other cells which perform EMT later on. This is possible, but we see that this is a very early event.
Thank you very much. I think that's all we have. And if you have any further questions, as you can see here, we have all the contact information. Please feel free to contact us. If you would like to, understand more about Professor Sabina Kazimierz-Bauer's research, you would like to contact her, you always can contact us first. We will pass on your request to her, okay? So with that, I would like to thank you all for attending this webinar. I hope you found it useful, and good luck in your research. And Professor Sabina Kazimierz-Bauer, thank you so much for joining us. Your research
thank you.
with our audience. I really appreciate it. Thank you.
Thank you, Wei.
Thank you. Have a good rest of the day, and good luck in your research. And to all the audience, I hope I can see you in the next webinar very soon. Thank you. Bye-bye.
Bye-bye.